Bismillahirrahmanirrahim. My name is Anisa Zibawan, and topic that I'm going to present is microbial fuel cell and its use in biosensors. So here is the table of contents. So if we look at the microbial fuel cells, so what are these microbial fuel cells? So they are actually used for the production of electricity using microorganisms, and they convert chemical energy into the electrical energy. So the microbes that are used in MFC technology are termed as exoelectrogen. So the oxidation reaction that took place inside the microorganisms in which food molecules are oxidized and electrons are produced. So these electrons are actually responsible for electricity generation, and the electricity produced during MFC technology is known as bioelectricity. So if we look at the background, then as we know that in a typical dry cell, battery works by generating electricity using chemical energy. So here in this case, electrons are produced at anode and they travel to the cathode, and at cathode electron undergo reduction reaction. So due to the movement of electrons, electric current is produced that powers up any device. So but in dry cells, electrons are actually getting absorbed in cathode, and its working gradually drops off due to the consumption of the fuel. But in MFC, as we have the bacteria, the process of cellular respiration continues as long as the food is available. So thus electricity generation from MFC is renewable process, and this property makes MFC more attractive. The microbial fuel cell technology was first presented by Michael Porter in 1911. So here we will look at the general setup of MFC and its working. So there are two chambers present in MFC which are known as cathode and anode. So these two chambers are separated by a special membrane known as proton exchange membrane. So organism that act as a biocatalyst is present at the anode. So if we look at the working, then biocatalyst at anode oxidizes organic substrates and produces electrons and protons. The protons are transported through proton exchange membrane, while the electrons are transported through the external circuit. Both electron and proton show reaction with each other as oxidized oxygen is converted into the water through reduction. So the conditions at anode chamber must be anaerobic, and bacteria at anode must be separated from oxygen. This is because oxygen hinders the electricity generation. So some of the commonly used microbes in MFC technology are Shivanella, Geobacter, Nostoc, and Anabina. So these act as a biocatalyst in MFC technology. So now I will discuss use of MFC in biosensors. So biosensors are actually the devices that can detect presence of biological analyte in the surrounding environment, and biosensors have applications in several industries. So MFC not only work as a power supply for biosensors, but due to their sensitivity, they itself act as a biosensor. So first we will discuss MFC as a power supply for biosensors. So for operating biosensors, MFC act as a long-term power supply. When we use MFCs in biosensor, then one problem that we face is low power output of MFC. So in order to solve this issue, we connect several MFCs in parallel or series connection so that the problem of low power output can be minimized. So when we are using MFC as a power supply, then external DC or external current is actually required here. So but the next thing that we will discuss is MFC as self-powered biosensors. So as we know that MFCs are highly sensitive to various environment and biological substances, so this makes them suitable to act as biosensors. For energy generation and sensing several factors, specialized MFC based self-powered devices are designed and that have no external electric power supply as in this case. So there are also several environmental factors such as temperature, pH that affect power and electrical potential of MFC. For example, if pH is changed from 6 to 4, then we observe immediate fall in the voltage. So this MFC setup can be disturbed by the environmental conditions such as pH and temperature, etc. So now we will look at the applications of MFC based biosensors. The first thing is biological oxygen detection. Biological oxygen demand detection. MFC based biosensors are used for the measurement or assessment of BOD of water. So biochemical oxygen demand is one of the important parameter for determining the quality of water, and BOD assessment based on MFC biosensors is less time consuming and efficient and is more accurate than conventional methods. And BOD measurement is dependent upon the proportionality of the current efficiency of cell with the concentration of oxidable organic substances present in substrate of MFC. For example, MFC based biosensors developed by Kim et al. In remote BOD detector and works over time period of about 5 years. And sensitivity of MFC based biosensors in BOD monitoring is affected by external resistance, dissolved oxygen concentration, and effluent flow rate. So this method is actually widely used for observing activity of microbes and pollutants in groundwater. And if the confined diversity of microbes and organic matter is present, then bacteria in MFC cannot carry out the process of oxidation effectively, and this is one of the main disadvantage of MFC. Now I will discuss MFC based biosensors for water toxicity detection. So detection of toxic substances present in water is important, so that necessary steps should be taken to make the water safer. So MFC acts as a best biosensor to perform this function. So MFC works in such a way that due to the presence of toxic substances in water, metabolic activity of microbes will get affected and consequently the substrate uptake. And as substrate uptake is directly related with the current output of MFC, so we can detect presence of harmful substances in water by monitoring disordered electric current produced by MFC. Type of microbes used in MFC is selected according to toxicant to be identified. Now some of the advantages and disadvantages of using MFC will be discussed. So advantages are eco-friendly, that during the process of MFC, no such a toxic chemical is produced that may cause pollution in the environment. So it is also a sustainable and renewable energy source. So for MFC technology, we use food and industrial waste as substrate, and this makes it more sustainable. Here, efficient and conversion of substrate to electricity took place. So if we look at the disadvantages, then it is difficult to scale up MFC on larger commercial scale. And during the working temperature, as we know that the working temperature also act as a hindrance for MFC technology, as microbial reactions stop at temperature below 20 degrees centigrade. And one of the disadvantage of MFC in biosensor is their low power output. So now I will discuss future perspective and current market. So the worldwide microbial fuel cell market was 8.6 million US dollars in 2019, and it is expected to reach 22.38 million US dollars by 2026. So one of the major thing that is going to happen in current era is that related to MFC technology is that US Army is working with Cambrian Innovation to test an MFC that can turn 250 liters of sewage into clean water and produce electricity to power itself. So the major area that is advanced in MFC technology is Northern America and China. So if we look at the conclusion, then MFC is the bioelectrochemical setup in which chemical energy is converted into electrical energy by a microbe. And MFC has a lot of applications such as in biosensors, bioelectricity production, and wastewater treatment. MFC are also used to design biosensors, and it is advantageous to use MFC because they are environment friendly. So here are the references. Thank you.